For our next session, Creating a Global Network, we actually have really great champions from the Angelman Disease Network from FAST joining us. And they have an incredible story, really, of building international collaboration. So I'd like to kind of have you work your way up. I did very short opening remarks for the afternoon, so we're going to start a little bit early, but that's just going to give us more time for Q&A as well. So, Megan Cross, FAST Australia Chairperson, Project Manager, and International Liaison for Global Angelman Syndrome Registry. And then Alison Berendt, DVM, Chief Operating Officer of Genetics Biotherapeutics and Chief Science Officer, Foundation for Angelman Syndrome Therapeutics, FAST, and Ed Weber back again to join us on this panel. So without further ado, I'm going to hand it over to the team here.
Thanks, and I hope you are not too carb loaded and you can stay awake for this next presentation. So I think that this really is a good story, and we heard a great story this morning. And this is really a good story as well, and it's a story of how this whole thing got about, how a parent advocacy group got together, pushed this forward, and how it really mushroomed out into something that was bigger than anybody could ever think of. But I'm going to step back a little bit and just give a little bit of a primer on me, where I came from, and Angelman Syndrome specifically. So what is Angelman Syndrome? It is it's a rare disorder. It affects this particular gene. What a beautiful molecule that is. This is UBE3A, a three-dimensional model of UBE3A. It's a ubiquitin ligase. It's an interesting molecule that the gene is imprinted, so it goes through maternal imprinting, which means that all of us right here in the audience, we're all producing UBE3A protein from the gene we inherited from our mother only. So the father's gene is turned down, it's almost completely silenced, but the maternal gene is the only one that's expressed, the only one that gets protein. You can get Angelman syndrome in a number of different ways. You know that we inherit one chromosome from our half our chromosomes from our mom, half from our dad. But if you have a deletion in the maternal chromosome, you don't get any UBE3 being made because the paternal gene is silenced. You can also have what we call uniparental disomy, which means that if you inherit, or if you through the process of the cells dividing, you get two paternal chromosomes. They're both going to be silenced. You can get AS that way. But what controls this, and it's really interesting that the cells have a way to identify what genes come from your mother or your father. You can have a mismethylation problem. So if the gene of that chromosome is mismethylated, it looks like the paternal gene. You can also get Angelman syndrome. This is what's considered to be a housekeeping gene. So UBE3A is a ubiquitin ligase, which means that it puts ubiquitins on protein. And this is kind of a molecular clock. So if you have proteins floating around in your cell, it bumps onto these ubiquitin ligases. It starts putting these little ubiquitins on. And if the chain gets long enough, if it's been around long enough, it gets a long enough chain to where it's degraded. And this ensures that no proteins are in your cell for a very long period of time. They want to be continuously renewed, continuously being active. UBE3A is one of these things that specifically targets proteins to put that ubiquitin on. And we've identified a lot of targets of UBE3A. None of them really are the smoking gun for what causes the disorder. So in all the things that I'm going to show you and all the research moving forward, we still are not really quite sure what the molecular mechanism of UBE3A deficiency is in Angelman syndrome. So I'm going to talk about the AS animal model, and this goes back, 1999, 98. I was a postdoc. I just got my PhD. I was at Baylor College of Medicine, and even though I was in the division of neuroscience, right next door to our laboratory was Art Beaudet, and he was the chair of the molecular human genetics department. And they were using a brand new technology at the time. This is 20 years ago. Brand new technology at the time to identify genes in the mouse model that were homologous to what we had in our genome. And then identifying if those genes were involved in human disease. And he had just found the gene that causes Angelman syndrome. And within a year, he had made a mouse model for Angelman syndrome. This mouse model showed the same kind of imprinting. So there was They just knocked out the mom's gene in the mouse, and no protein was made in the kids that had that mutation in that gene. These animals had seizures, which is very close to what we see in children with Angelman syndrome that have a high propensity for seizure. They had a motor coordination defect, and we see this in the human population as well. This is a accelerating rotor rod where you put the animals on, you see how fast they fall off. And then if we looked at the cognitive aspect of these mice, they were disrupted as well. And we could even look closer at their brain and see a difference in their synaptic function. So the animal model was a perfect model and one of the first models for a human cognitive disorder back 20 years ago. So I spent a lot of time with this mouse model, and when I left Baylor and started my laboratory at Vanderbilt University, Art let me take the mouse model with me to do the research. And I think this is, you talk about those big people in the field. Art was one of the biggest, and he was a guy that had tons of money, huge in the field, had spent a lot of money making this mouse model, and he freely gave it to anybody who wanted to do research with it, which was, I think, at the time, really important. Now, the other aspect of the importance of this animal model is this is a brain-specific disorder. So you can't get samples. It's really hard to get samples like you would in other disorders. So the animal model really became central to the research that we were doing. So because it targets proteins for degradation, we wanted to look and see if there was changes in the protein anywhere, and we looked everywhere for biochemical changes. I maybe spent a year and a half looking at all the different types of proteins that are expressed in the brain. We couldn't find any differences. But at one point in time, we found a single difference in a protein called calcium calmodulin protein kinase 2. I don't expect you to remember that. We kind of shortened that up into CAMK2. And there wasn't a change in the total amount of CAMK2, but there was a change in its phosphorylation state, which made it inactive. So the little phosphates, they modified the protein. We saw a lot more of these phosphates on that protein, and that protein was basically not active in the brains of the Angelman syndrome mouse model. So we decided, how the cool molecular identification, but really what are you going to do about it? Well, there was a researcher at the time that was going and mutating those specific sites on the CAMK2 because he wasn't interested in Angelman syndrome, he was interested in CAMK2. And so he mutated the sites where those phosphorylation sites were unable to accept that phosphate, okay? So here we had our AS mutant mouse. We had this CAMK2 mutant mouse. So ours had too much of these phosphates. This one couldn't put phosphates on. So we had to make a love connection here. I mean, we had to get these mice together. And what we were hoping for was that the progeny of these mice would basically have some of those sites that would allow the enzyme to remain active. Very, very simple experimental design. When I was first starting out as an assistant professor, and I think the assistant professors here could probably appreciate this, the mentoring committee at Vanderbilt were really not enthusiastic about me trying to rescue a mouse model with a mutation by breeding it with another mouse model with a mutation. So I couldn't even get a graduate student to do this work. And so I had an undergraduate from Meharry come over to Vanderbilt. She didn't know any better, so I put her on the project. We bred these mice together, and we saw immediately that there was something different about these mice. Now, for the scientists out there too, you can relate to this. There are certain times in your career where you identify something that you know the implications of. It's going to be seminal, it's going to be huge, and you're the only one in the world that knows that. I mean, this is really what drives us as scientists. And we knew that there was a difference in those mice. We were able to recover all of those symptoms that we saw in that AS mouse model. We genetically cured that mouse. And so I published the paper and made a big splash. And then the local media picked it up, and they did this. I know I look pretty young there. They did this nice article about how we genetically rescued these mice. And then something extraordinary started to happen. We started to get, and I started to get emails from parents, not in just the US, but all over the world. They were asking, what does this mean? If you can cure it in the mouse, how come you can't cure it in the person? What are you going to be doing next? I had done the experiment. I was going to go back to doing my Alzheimer's disease research. AS research was really not in my realm. I had no funding for this. But really, this got the ball rolling. And so I decided to present this data at the scientific conference in St. Louis. I met Terry Jo for the first time when I was at Vanderbilt, and she has a child, the first child I ever met with Angelman Syndrome. But what happened was I met also Paula Evans. And Paula Evans, she had a child with Angelman Syndrome. She started asking me very pointed questions about, what do we do next? And what does it mean? And what is it going to take for you to come up with some kind of a profound treatment for Angelman Syndrome? And so I told her, well, first thing it's going to take is a lot of money. It's going to take a lot of money to move this research past this animal model. I said, and the other thing that it may take is somebody who has a child with Angelman Syndrome, a senator, a governor, an actor or actress that everybody can relate to. And that pretty much set the stage. So the Foundation for Angelman Syndrome Therapeutics was started. God, I even look younger there. So this is at the University of South Florida. After the foundation, I think this was the culmination of the foundation, were these 4 members. They came down to look at the lab, look at the research, and say, okay, what is going to be next? And with that, the story will continue.
So this is just a little video to help introduce Angelman Syndrome and give you an idea of the impacts of Angelman Syndrome, and it's featuring our founder, Paula's daughter.
Ainsley was 10 months and 1 day old when we were told she would never speak, that she may never walk, that she would have debilitating, life-threatening seizures, and would never learn to read, go to college, have a job, or live on her own. They told us Ainsley would forever be dependent on others. There were no treatments for her, no hope for a cure. Ainsley, like thousands and thousands of children around the world, has Angelman Syndrome. The first 2 years were especially difficult. Like many of her friends with Angelman Syndrome, Ainsley does suffer debilitating seizures and numerous health problems. But at 2 years, 8 months, and 8 days old, Ainsley learned to ride a tricycle. At 3 years, 10 months, and 9 days old, she took her first steps. At 5 years, 2 months, and 3 days old, Ainsley signed her first word. 6 months later, she learned to read. Ainsley isn't the only one proving the doctors wrong. In 2007, Angelman Syndrome was cured in the mouse model, and research towards a cure for AS has been moving forward ever since. Angelman Syndrome will be cured. The only question now is how many more days, months, and years Ainsley and her friends will need to wait. How many more days, months, and years before Ainsley speaks her first word. The doctors were right about one thing. Ainsley would be dependent on others. Funding the cure for Angelman Syndrome is up to us, and our children are waiting. CureAngelman.org. Raising hope through research, changing lives together.
So the way the FAST US board was structured had a board of directors sitting at the top. 99% are parents. There was just one non-parent, all-volunteer board. There was a scientific advisory board that Paula had brought together with the help of you, Ed, constructed this. Pretty much like what we heard this morning, going out and looking for those specialists to come on this board. And there was a clear, concise mission to bring treatments to our kids as soon as possible. By chance, a venue for a gala in Chicago became available, and Paula pulled it together in typical Paula fashion as quickly as possible, had a great attendance, and it was something that spearheaded the fundraising efforts for the foundation. This is my daughter Molly, and this is where I come into the picture. Molly was diagnosed with Angelman Syndrome 3 weeks before Paula, actually 3 weeks, I have to remember it. It was around 3 weeks, I think, before the foundation started in the US. So I'd made connection with Paula through, some of you might be familiar with listservs, but before Facebook, it was a lot harder to connect with people around the world. Facebook's made a huge change for us families of rare diseases. And I had a situation where, probably not unlike a lot of you, I knew something was wrong with Molly. I'd hit up Dr. Google, and I'd tried to convince people to test Molly for Angelman Syndrome. So by the time she was diagnosed, I wasn't as devastated. I was happy that I'd finally had a result for her, and I really wanted to know what I could do going forward. So I had a few different things in my mind of what I could do or what I really wanted to do. My background is in information science, so I really wanted to collect information on Angelman syndrome in what we thought of then as a database. I was looking further and further into Google, looking to try to find that information about Angelman syndrome, and there really wasn't a lot of information out there. And within Australia, there wasn't actually a community for Angelman syndrome, so I wanted to do that. I wanted to build that Australian community, and I wanted to sort of look at what there was in the rare disease space as well. Around this time, I'd found the article online that Ed had written, so I was one of those parents that had sent an email to him in the middle of the night, and I was overjoyed when he responded very quickly to that. I had to work out how I was going to get some money to bring Ed out to Australia and tell these families what was happening. And around that time, I was aware of Rare Disease Day. It was in its second year. So we held a local community walk, and we got people in to walk to raise some funds. The event's now, the next Rare Disease Day will be its 12th year, and it's grown to one of the largest community fun runs and walks. So then we invited Ed for his first visit out to Australia, and we were able to bring the community together as well as medical professionals and researchers to hear about this discovery and what that meant to Angelman syndrome. And from there, one of the founding board members is there, Jen, with her daughter. What I also loved about doing that is we had a lot of older families who'd never had any hope for research. They'd given up. And we were able to bring people from all age groups and from all across the country together. What we needed then was our own scientific advisory board. So we continued to hold these sessions, bringing out the experts in the field into rooms in Sydney, Melbourne, Brisbane, so we could build a scientific advisory board of our own and build our board, and also build our community, because we need the community, of course. Meanwhile, things were growing at a rapid pace in America, and this is what the annual fundraising gala looks like now. Things like the paddle raise, they give you goosebumps, you know, when you're sitting there and you see someone raise their paddle and you look around and they're a researcher or a pharma company practically giving back into their own programs. It's really exciting. But what that did is it ended up giving us an opportunity to launch into our new research environment. And I'll pass it over to Ed to talk about that.
So I kind of touched upon this earlier about having different perspectives come in and looking for collaborators. And so we, in collaboration with the foundation had extra money. We went and actively searched out different researchers to be part of this consortia of researchers to do Angelman syndrome research. So I worked with the foundation. I had already identified a few people that I wanted to look at. I'm classically trained as a biochemist. So we wanted to have an MD involved. So we asked Dr. Anderson to come. She was a pediatric epileptologist. She knew a lot about EEG. She knew a lot about seizure control. And we really wanted that clinical kind of aspect. Dr. Dindo he worked in Art Baudette's lab, so he was trained with the person who identified the gene, knew a lot about Angelman syndrome, but he was a geneticist, so he could bring that kind of genetics kind of aspect to it. Somebody said reaching out to somebody who had a specific technology and bringing them in. Dave Siegel was at the UC Davis. He was working on what was called Zinc Finger, which is a gene editing tool, and he was not interested in Angelman syndrome whatsoever. And we knew that could actually be used to turn on that paternal gene. And so we brought him into the fold and said, okay, you need to refocus your research and the technology that you have into this particular disorder. And so you can see the utility of having different people looking at this from different perspectives. And then we would have weekly meetings showing data in real time. So you don't have to wait for a paper to be published and go, oh, this is a cool discovery. You already knew what was going on in these different laboratories. And this has since grown. We have other scientists that are involved in this consortia, or at least we did before I left. So it's gotten larger and larger. And so you have more and more perspectives, and it really drives the research. And again, doing this and sharing tissue, sharing results, sharing reagents, it really made the research team much more effective. And this was a result of that increase in the advocacy and increase in the fundraising.
So this is where I come in. So I come in on the backs of a tremendous amount of work in 2015. So this had been going on since 2007 when Paula first contacted Ed and then Megan got diagnosed. So my daughter, her name is Quincy, and she was diagnosed in 2015 when she was 5.5 months old. And I'm a veterinarian by training. I do clinical research, and I discover and test medical devices. So I do a lot of research and design clinical trials for animals with naturally occurring disease. And so for me, it was my whole career, which was at the University of Pennsylvania, actually, so it's great to be back in Philly. My whole career was really about understanding what's possible and impossible. And my husband and I, we work together, we do the same thing, and we both always had this motto when we started doing interventional radiology that things are either possible or impossible, and there's no in between. And this was always a motto that we had. And so when we had our first daughter, she was neurotypical, everything was fine, she was great. I got pregnant with my second daughter, and when we noticed that she was having delays and that there was definitely something globally wrong with her, and everyone around us told us she was normal and there was nothing wrong with her, we pushed very hard for some genetic testing. And we actually had to pay for it out of our own pocket because we were told that it would never be covered by the insurance company. And when we did that, we got a phone call about 2.5 weeks later, so she's almost 6 months old at this point. And at that time, we brought her to the neurologist right before that, and he said, look, she's catching up, she's doing just fine. Worst case scenario, your daughter has a little bit of cerebral palsy. So if you could only imagine someone saying that to you as the mother of a 5-month-old little girl, that your child, don't worry about it, it's just a little cerebral palsy. Well, for those of you or anybody that has cerebral palsy, that's not benign, and that's not something anyone would choose or want to live with. And so we heard that, and we were devastated, but we get a phone call, and on the phone, the neurologist says, Miss Weiss, that's my married name, I have some catastrophic news. It's on the phone, and he says, your daughter has Angelman Syndrome. And I'm a clinician, I'm a doctor, I understand what diseases are. And imagine if you were a parent who doesn't even know what syndrome means. And my answer was, can you spell that? That was my response to this man and my 5-month-old daughter. So he spelled it, and I'm on the computer because I'm in a coffee shop, and I, of course, came up with Wikipedia, which is probably the worst thing you can ever do, is search something on Wikipedia that is life-changing for you and your child. And so he continued on about Angelman Syndrome, and he told me all the things she would never do. What's her prognosis? Well, she's never going to walk, she's never going to talk, she likely is not going to have friends, she won't go to a typical school, she's not going to go to college, she's not going to get married, she definitely won't have the life that you or I have, but she will be happy. And you should love her and let her be happy. And this all happened in like 30 seconds. If you could only imagine a 5-month-old little girl being doomed by this news. And so, of course, my response first was to fall to the floor and cry, and that happened. And then I started asking, well, what will she be able to do? And I was told all the things she won't do. And then I thought, well, will she die from this? Okay, so these are all the natural things of mourning, right? But this happens in like 1 minute and 35 seconds. So she's not going to die from this, but she can't do anything. So now what's next? And then the next thing is, well, if I give her the best life, what could she be capable of? So these are the things it's saying she's not capable of, but what can we provide for her? What could she be capable of? So then we went through that process. How do I teach her and the world to have confidence about her potential and to not treat her as somebody who can't do anything? So these are all the things that parents go through as they get these diagnoses, and for some people this takes years. For me, it took about 15 minutes to go through all of this in my head. And how do I get her to live the most independent life possible? And this is where I was going, and what I realized at that moment was that I had one thing I had to be, and that was her advocate, and nothing else could change. But then, I'm a scientist, right? I'm a clinician. So I started reading everything. I PubMed everything. Anything that had the word Angelman Syndrome, I would read. And one of the first articles I came across was this article that looked at individuals with Angelman Syndrome that have a mosaicism, which is very, very rare. It's like 1 in 3% of kids with Angelman Syndrome are mosaics. And what I found was that kids that have only 1 to 3% of their cells with a functional gene, where my daughter has 0, 1 to 3% of cells and of neurons that have functional UBE3A were a different child than I was just doomed to live with, and that she was doomed to live with. And so I realized it may not take a lot of UBE3A for her to be a different kid and for her to have a better life. And so kids with 1 to 5% of UB3A, they walk normally. They have no seizures. They have some speech. They maybe have a little bit of a balance issue, but they're not falling all the time and hurting themselves. And so that was hope. And then 20%, oh my goodness, this kid could almost be normal. This kid can graduate from high school. This kid might be able to go to college. This kid's gonna have friends. This kid can have a relationship. These are all the things that are going on for a 6-month-old little girl in her mother's head. So I came across this, and I realized that we needed to give her back even just a little bit of UBE3A, and that might make a difference. And then the next paper I read was Ed Weber's paper, where he treated mice with Angelman syndrome with gene replacement therapy, and adult mice had improvements in their cognition. And all I wanted for her was to have some cognition where she would live a happy life and an important life to her. And that's all I wanted. So I started calling everybody. 1 week after her diagnosis, a paper came out that said that you can actually activate the paternal copy of the gene by unsilencing it. What? So suddenly, things I'm reading are coming out, and it's coming out quickly, and this is 2015. So now I realized it was time for me to be a clinician, and I had to understand Angelman syndrome if I was gonna help figure out a way to fix it. I had no idea what was going on with the FIRE team. I had no idea what else was out there other than what was published on PubMed. So I started reading everything, and what I realized is that you can replace the gene, like Ed did, with a viral delivery, or you can activate the paternal copy of that gene by unsilencing it or inhibiting that RNA with things like artificial transcription factors, like Dave Siegel, or antisense oligonucleotides, like Art Baudet was doing. So you can stop the stop. Or there are downstream therapeutics that you can use where you can actually try to help the synapse. It's a synaptopathy. So you can fix the synapse and all these accumulated proteins, and you can try to fix that. But the one thing I realized very early was that this was non-degenerative. So earlier is probably better, but it is non-degenerative. And I felt like we had some time to do this right, but not a lot of time. And so that was when we had to make a decision on what we were gonna spend our time as a family doing for her. And so we needed to make sure that people were prepared. What animal models do we need? What outcome measures and biomarkers do we need? What is going to be important for clinical trials, and how are we going to get these scientific discoveries from academic labs into the hands of people that can translate it for humans? And so when I read all of this research, one thing I realized was it is an awesome time to be a mouse with Angelman syndrome. But I'm not confident if it's awesome to be a human with Angelman syndrome. And what I realized is that she's not a mouse. So I started calling everybody I could. I called Art Baudet. He got on the phone with me 48 hours later for 2 hours. I called Ed. He responded to my email. I called Jim Wilson at the University of Pennsylvania. In 45 minutes, he responded to my email. I called Dave Siegel at UC Davis. Anybody that published anything, I called and said, what can you do for my child and for all the other children living with Angelman syndrome? And what I realized was that mice are lucky, humans are not. But I also realized was that if we did it right, we might be able to make a difference in the short term for these kids. So I went to a scientific conference and I listened to all of these people talking about Angelman syndrome and all the amazing research that was on the backs of everything that I've just learned in the last 3 weeks, that they've been doing this for years. And they had great funding by the NIH, and everyone in this room is rubbing shoulders about all the R01s that they received and how much funding they got from the NIH. But what was missing was, what was that funding doing for a human with Angelman syndrome? They were asking questions in mice, they were answering questions in mice, and then they were asking more questions in mice to answer them more in mice. Funding was going to mice, and it was not going to humans. And there was this valley of death that was very clear no one was leaping forward to. And why? Because these brilliant scientists didn't know how to take that leap. That's not how they were trained. Very few scientists were willing to take that leap and join forces with a foundation to say, what can we do to move this into human medicine? And Ed was one of the very few scientists that was willing to do that. And that's where it was groundbreaking for this foundation. And so I couldn't wait for a treatment, but I had to help prepare for one, and I had to help create one. And so after I went to this conference, and I stood up at this conference, and I said, why are we not translating this for humans? And all of the researchers looked at me like, who is this person? And this is why we don't want parents in this room, because parents shouldn't be saying things like this and criticizing our brilliant work. That's when Paula Evans called me, and she said, I would like you to be the chief science officer for our foundation. And I would like to help you bring on that translational piece that we're missing. And it was hard for me to make that decision. There's other foundations in the Angelman space. I didn't want to commit to one. I wanted to support them all. But I realized this is an opportunity that I can make a difference for Quincy and for other kids living with Angelman syndrome. And so it was during that time that I became the chief science officer. And this consortium of scientists, I started going on these calls, these weekly calls. And what I realized was, holy cow. They are doing so much for Angelman syndrome, and I had no idea because it wasn't published. Why? Negative data doesn't get published. Why doesn't it get published? It does in veterinary medicine, so this was new to me. It doesn't in human medicine. It doesn't in science. No one cares if it's negative, so let's just waste more money and let other people do negative things over and over and over again so that they don't know that it didn't work. But these guys, they were talking about negative data. They were talking about positive data. They weren't repeating the same things over and over again. And I was so much more hopeful that there was so much more going on for this space. And this was because they were working together, and that was super important. And what I realized was that as a foundation, if we de-risk, then we are more appealing to pharmaceutical companies. We need to put in, we need to de-risk it, we need to show it's valuable, and then people will come in and put more money into it. And that's where we were gonna focus our funds. But foundations have limited resources, and this is all on the backs of parents that have to pay for these kids. But these kids are super expensive. So what do we do with these expensive kids that we're paying for therapies for, but we're trying to fundraise to fund research? But whose job is it to fund all of this research? So this is a really big problem for foundations and advocacy groups. And so the families put their efforts together and they fund research. And so what happened was that I happened to be really, really good friends with another parent of a child with Angelman syndrome. I didn't understand where he was from, what his means were, what he was about, other than he had a child and he lived in the UK. And we talked on a monthly basis just about the research. Now as the CSO, he wanted to know what was going on. So I would give him updates on things as they were, as they were happening. While new papers were coming out, I would explain them in lay terms to him. And he came to New York to visit me, and we had lunch with him and his family, him and his wife. And he's like, what do you need for a cure for Angelman syndrome? And I said, well, my first gala, I was at this fundraising event for FAST, and I turned to Ed at the end of this event, and we were having beers at the big bar. Hopefully you remember this. And I turned to the whole fire team, there was 5 of them, and I went to you first, and I said, this was an amazing event. It was my first gala. I'm from New York City. I'm a complete fundraising snob. We raised $3 million in 30 minutes in New York City. This was the most beautiful event I've ever been to. It far was superior than any event I've ever been to in New York City. I work for a not-for-profit, so in New York City, I know how to raise money. How can we only raise $400,000 tonight? I don't understand how we didn't raise $3 million tonight. This was an amazing event. And I said to you, how much is it going to cost for you to cure Angelman syndrome with AAV gene delivery in 24 months? And you looked at me and you said, ugh. And I went to the next person with ATFs. Yeah. What is it going to take? What is it going to take? What is it going to take? And they said money and people. And I said, well, people are money. So I need a budget from you in 24 months for a 24-month plan on what it's going to take to get your gene therapy to an IND, for IND-enabling studies. And I went to the ATF guy, and I went to the ASO guy, and I went to every guy or woman, and I said, I need a budget. So in about 3 months, we had budgets from all of you for a 24-month plan. That 24-month plan was $5.8 million. That 24-month plan was for every approach to be in IND-enabling studies in 24 months. We had advisors that came in, what do you need to do that? Because not everyone knew what to do, because that's a hard process. And so I went to my friend who sat me down at lunch and said, how much is it going to take? And I was like, oh, it's going to be like $5.8 million. And he's like, I have $5.8 million. I was like, you have what? And like Ed said, it takes one rich family. It takes a senator. It takes an actor. It takes a famous person. It takes money. It takes awareness. And they made a donation for $5.8 million to the Foundation for Angelman Syndrome Therapeutics, and every one of these programs started on a 24-month clock. And I was nothing more than a project manager to say, do not call me if you're late with results, only call me if you're early. And that was our 24-month plan. And basically from there, we had a huge influx of excitement. And we launched an initiative during that year. So that next year, we launched the Angelman Syndrome Biomarker and Outcome Measure Consortium. That was a consortium of 2 foundations, as well as pharmaceutical companies. Terry Jo Bichelle, we brought on as the director of that consortium. And we started looking at pre-competitive space for developing biomarkers and outcome measures for Angelman syndrome, because remember, that's what you need for clinical trials. And so Ed helped us develop that process. We brought in Agilis Biotherapeutics, which is now PTC, and they helped us to kind of develop how to do this for pharmaceutical companies and for industry. And between all of the money going into research, between the funding and the support for the Outcome Measure Consortium, we were on the map. And suddenly we went from 2 interested pharmaceutical companies. In 1 year, it was 7, and in 1 more year, it was over 10. And Angelman syndrome was finally on the map, and we were looking for meaningful change. And so we had these biomarkers and outcome measures that Terry Jo put together as a really great way to look at all of different things that are focused on Angelman syndrome, what's alike and what's different, and what are important domains that are meaningful for families and for kids with this disorder. And we had these domains. We did a disease concept model, and we were able to interview families and say, these are the order of domains that are most important. And now we need to come up with outcome assessments that are appropriate for this population of individuals. And now that's the process that we've done over the last 3.5 years. And so sleep and epilepsy, communication and learning, motor skills, those are all super, super important. So the next year, we had our next fundraising event, and we had just brought in all this money. People were excited. There was so much discussion about how we can now actually cure Angelman syndrome because we have money to do it. And we have a plan. We have a roadmap to a cure for Angelman. You need an ask. If you don't have an ask, you don't have money, you don't know what you're asking for, no one's going to give you money. And so you need to understand your path forward. And we had that. And now we had our biggest fundraising event of the year. And that year, we went from $400,000 my first year to $1.2 million my second year. And then my third year, as part of the foundation, we raised $4.4 million just at that night, just as a paddle raise at that event. And what we realized was with passion, vision, and money, there's a lot of things that are really possible.
It's worth saying a celebrity came into the mix as well.
Yes, and so one thing that we didn't mention was that when Paula and Ed first got together, Colin Farrell's son was diagnosed with Angelman syndrome. And so that certainly helped because Colin Farrell brought a lot to the table about what this is in the public domain, the fact that you don't have to be scared of disability, the fact that this is a very rare disorder, rare disease happens, but you can live with Angelman syndrome. And he really brought a lot of awareness, and he brought a lot of excitement for families because they got to come to this event every year and meet Colin Farrell. And he would talk to every family, he would hug every parent, he would talk to their kids, he would hug their kids. And suddenly this was something that was really taking a lot of space in the public media, and that was very important for the Angelman community as well.
This is a fire video. Do you want to?
Yeah, do you want me to show the video? This is just a quick video where when all of this money came in, this was really an interview of all of the scientists that were involved in Angelman syndrome and what their perspective was on the future for this disorder.
When the US government decided that they wanted to go to the moon, they started by putting a team of scientists together and gave them a common task of how would you get to the moon. It was a goal that people might have thought was impossible, but with the right resources and the right team of scientists, they were able to achieve wonderful things. And the FIRE team is a lot like that.
The Fast Fire team is a group of scientists from diverse research backgrounds that work collaboratively to find a cure for Angelman syndrome.
Each of the members has been trained in a very different environment and a very different discipline, and so we all have unique perspectives on the way we approach science, and I think we've made a lot of gains and progress because of the feedback that we get from each other on almost a weekly basis.
The more science starts to look at cognitive disorders in general, we find that there's a lot of overlap. This is profound. This means that any treatment that we come up with Angelman syndrome could be expanded into a lot of different cognitive disorders, including Alzheimer's disease.
Getting the opportunity to interact with the families and the individuals is probably the most rewarding aspect of what I do.
You have to understand that as a scientist working in a laboratory with mouse models for all these other disorders, a lot of times you may never meet anybody who has that disorder.
You can see what effect you would have not only on the patient but on the family. It gives you a drive to keep going because you have a connection now to the goal that you're trying to achieve.
Exciting developments and discoveries that have been made in the last 3 or 4 years are directly due to the support of the parents. So, in essence, I'm working for the parents of the children with Angelman syndrome that are supporting FAST and supporting the research that FAST scientists do.
Cure used to be a 4-letter word that people didn't really want to say because they didn't want false expectations. But now it's a reality. What we really want to do with that and how we think we can get there is by helping to fund the science that we already know works.
We have the goal. We know what to do. There are 3 ways that you can come up with treatments for this disorder. You can either turn on the paternal gene and reactivate it. You can do gene therapy or protein therapy, or you can come up with therapeutics to overcome the absence of that particular gene. So that path is clear, and more funding would just make that journey shorter.
I really think there is a feeling of hope and a reason for hope. I mean, there's so many things happening now in this community. I mean, with the research that's being done, with the pharma companies that are coming in, I mean, there's a momentum, and the momentum keeps building.
I believe that a cure is on the horizon, and we are working hard towards this goal.
I am very confident that we will discover or develop a new therapy that treats Angelman syndrome. I wouldn't be doing this if I didn't think that we could.
I think that really touches nicely on one of the previous panels where we were talking about connecting with researchers and how important it is for families to connect with researchers. Alison and I both see it, what an impact our kids have made on researchers. And we know researchers and pharma or people involved in pharma that have left the community still want to connect with us because they're still invested in our journey. Whoop. And this is just a picture of the first ever Angelman syndrome biomarkers and outcome measures group. From this consortium, it connected a bit with my vision that I was thinking earlier back in the piece when Molly was diagnosed about collecting information for Angelman syndrome. And we had been working on that project. And in a way, it was a blessing that it took some time to get together. Back when we started talking about it, I think it really freaked out our ethics. Our ethics is what we call the IRB boards in Australia. So our IRB boards were, you want to collect health information online? It took them a long time to think about what that might look like. But the forming of this group really helped be the catalyst for what this global Angelman Syndrome Registry was. We were hearing from the FDA how important natural history studies were in drug disease development for rare diseases. So we were able to think of what that project might look like. We had some ideas and we were working on some of them, but as I said, it was a moving target. We were changing as we went along. We knew we wanted it to be online. We wanted it to be caregiver initiated. Our children can't speak, so we needed to be their voice in this instance. We wanted it to be inclusive, so it wasn't about where you were in the world or your socioeconomic status and whether you were able to attend a clinic. It's modular, which I'll go into a little bit later. It's global. And we had very strict ethics and privacy, and they're also a moving target, as you will all know. It needed to be accessible. The data and the information in that needed to be able to go to whoever wanted it. There was a study at the time, back when we received our diagnosis, being done in the United States. It was a natural history study. You had to go back, I think, annually for 5 years. We seriously considered being involved in that study. I'm glad we didn't, because this is what it would have looked like on a plane for 26 hours. And I don't think anyone else would have been happy on that journey. So this is going a little bit into the modular sections of what we collect. This was based on that natural history study that was done. So we didn't want to reinvent the wheel, we wanted to complement it. So these are the sorts of domains that we collect information in. And this touches on the reality of rare disease. I think these statistics will be the same no matter what disease you're looking at. The majority of the population, unless it's specific to a certain ethnicity, would reside in Asia when we think about these things, we need to think globally. Consents. So the way, I think this has been touched on a little bit today as well, how you consent people collecting your information or how you ask your participants to consent is really important, both for the legal eagles that sign this project off, but also for parents being able to be in control of their information and how it's used. All requests for data go to, this is an example of how it works. So all the requests go via the data curator. We've got a curator that sits part-time in Brisbane. You give permission to them before you can share your data out, and no one has access to anyone's identifying information without their consent. So the parents or participants actually sort of have a two-step process before anyone sees your information. I'm conscious of time, so I'm actually going to skip past that one. This is the sort of thing we give back to the community. We give them information, statistics, so that they can actually be rewarded for their participation. Things that you would expect the community to ask, how likely is it that my child's going to have seizures, and when might that happen? For clinicians, it's interesting to understand what might have led to their diagnosis. And because communication is such a huge aspect of our kids' disorder, this is quite a complex graph, but it's helping us understand what types of communication our kids use. What we're working on now is annual reviews. It's a longitudinal study, so we can track changes over time. Clinicians being part of the process, translations to make it truly global. We have 5 translations almost finished in Chinese Mandarin, Italian, French, Spanish and Portuguese. There's actually 2 more being translated at the moment. Everyone's on their phone these days. That wasn't like it when we first started this project, so we've got to convert that over to a more mobile phone-friendly format. Apps, there's been some discussion about Health Kit today as well, looking how we can draw in wearables and things like that, or hospital reports from apps. Research partnerships, the data's open to everyone. We just have to go out there and ask people to be involved. How is this going to end up answering some of the questions that the ABOM have, what we would like to know there, and be ready for clinical trials.
So I'll go through this really quickly just so we have some time for questions. But with all of this, all the data, the Outcome Measure and Biomarker Consortium, all of these researchers really working together in order to develop meaningful therapeutics and not being competitive, but being pre-competitive and supportive of each other, knowing that they all played a role in every potential therapeutic platform that's being developed, more big changes happen. So I wasn't kidding when I said I gave them a 24-month timeline, and that's exactly what we did. And so about 17 months into that 24-month clock, we got a text message from one of our researchers who showed us this graph. And basically what he showed was in human iPSC, so in human neuronal cell lines of Angelman syndrome, he showed that with a human antisense oligonucleotide, he was able to completely knock down the paternal copy, the paternal antisense transcript and activate the paternal copy of the gene by 100%. And that was more than anybody had shown in the past. It was more robust, it was quicker, and it was at a lower concentration of drug. And he showed this to us, and he compared it to many of the other ways that it had been done. And as I had told you, 1 week after my diagnosis, Art Baudet had published a paper showing that you can rescue adult behaviors in Angelman syndrome with this same approach using the antisense oligonucleotide in mice, and that you can activate the paternal copy throughout the entire brain. And so when we realized we had this, and we knew that there were other pharmaceutical companies interested in this, but they had been interested in it for 5 years, and they were ready for clinical trials in 2 years, every year for the last 5 years, we knew that we were still probably 2 to 10 years away from the 2-year mark every 2 years for the last 5 years. So we felt like, you know what? Maybe we need to think bigger than that one pharmaceutical company or 2 pharmaceutical companies. We need to think more globally. So we decided as a foundation that we were going to license this technology from Texas A&M University, and we were going to start our own biotech company. And we were going to de-risk this further so that we can be singularly focused on one disease, not the 50th in the pipeline of neurogenetic disorders. And we could bring this forward for a human clinical trial and de-risk it so a pharmaceutical company would be more interested in taking it. If we put the money in and we de-risk it, maybe we'd have more success. So in 2017, FAST launched Genetics Biotherapeutics with FAST and Texas A&M University and some angel investors. And we basically created a for-profit entity out of a not-for-profit. FAST was the majority shareholder, with the ultimate goal, if this was at all financially successful, which is obviously not our motivation. Our motivation was a treatment for our children. If it was at all successful, FAST would become financially healthy so that they could fund more research in more areas, not put all their eggs in one basket, and ensure that no patient gets left untreated, that there are patients' assistance funds, that we can ensure that people that don't have insurance will not be without treatment, that we can ensure we can support families in a much grander way without begging parents for every dollar at every gala event. And so we decided to de-risk this in monkey studies, and we went right into the non-human primate because non-human primates have a conserved sequence to humans and rodents do not. And from there, we were able to basically take a program from first IND-enabling studies into last IND-enabling study in less than 16 months, which is basically 1/10 the duration of time it takes a pharmaceutical company to do the same thing. And we were able to do that by surrounding ourselves with experts and knowing that we need to meet timelines, we need to do this well, and safety is our #1 priority. And we surrounded ourselves with amazing people, and we partnered with the FDA. The FDA was huge in supporting this program because they wanted to see this be successful as a model for other rare diseases, and they knew we did not have the resources to do it like a standard pharmaceutical company would. And so we had so much support to be ready for first in human in 2020. And so we basically took a vision that Ed had, and we brought this forward for actual human translation to this is only one example of the many examples that the foundations have helped to fund in order to not fall into this valley of death of no translational medicine because we don't know how. And so we were able to do that because we had the funds and we brought in the expertise to do it safely and incredibly quickly. So the roadmap to success for us is having the combination of vision, passion, and just a little bit of naivety. If you're naive, you don't know any better than the fact that it's possible. And remember I started with saying things are either possible or impossible and there's no in between? Well, until you show us that it's impossible, I'm pretty confident it's possible. And if it's possible, we're gonna keep going until we can prove to everybody, especially our children, that this is possible because we are their only voice. And the vision of our foundation is to help every single one of these kids. 1 in 15,000 kids are living with Angelman syndrome. That's potentially 500,000 kids in the world. We want to help every single one of them, not just the ones that could afford millions of dollars of gene therapy or gene-modifying drugs. And so it's very important for the foundation. So where we started when I joined FAST and where we are now is very different. And the way it looks today is that there are 5 people looking at gene therapy. There are 3 people looking at antisense oligonucleotides for paternal activation. There's an artificial transcription factor program. There's 2 CRISPR programs. There's multiple downstream therapeutic programs. And these are all the pharmaceutical companies that are now involved in Angelman syndrome because we had a vision, we organized it, and we were able to hand them on a silver platter a beautiful disorder that they can help treat. If you de-risk it, you make it much more appetizing for pharmaceutical companies that don't want to waste money starting from the beginning. And that's what we really tried to do, and this is where we are now. So we are across the finish line in clinical trials in some disorders and in some platforms, and we're actually approaching clinical trials this year in many others. So we want to thank you certainly for your time and for listening to our story, and we would be happy to answer any questions.
Questions?
Hi there, my name is Candace Lerman, and I have a rare disease called ITP, or immune thrombocytopenia, which we have a naturally occurring model in canines, which is something I've been very interested in because I lost my dog to a brain tumor a little over a year ago, and we donated it to science. So I got introduced to the translational model world through naturally occurring models. So my question for you is that I'm looking to increase research into ITP because none of the FDA-approved drugs work for me, so I use an off-label chemo treatment. How difficult is it to approach a researcher because there is a naturally occurring canine model and suggest to them to do this research, given your background on both sides of this issue?
You know, I think that my experience has been, so I'm approached all the time by parents of dogs and cats who have really rare diseases or really bizarre anatomical abnormalities that want a medical device developed because they think they can reroute things. I mean, I just do really interesting plumbing in animals. And so I'm approached all the time about things like this. And veterinarians in general are incredibly compassionate. And so I'm always on this side of the conversation, someone asking me how I can help their pet. So when I went on the other side of that conversation and I was asking someone how they can help my pet, but her name is Quincy, the receipt I got of my inquiries was phenomenal, was warm, was expedited. There was nobody that didn't reply. There was no one that didn't answer back. And veterinarians are even more compassionate traditionally than basic researchers, no offense, and then physicians as well, because they go into veterinary medicine because they don't want to make money and they want to love on animals. So they have that personality. So I think it would be very easy. And I think you just have to contact people that are published in immune-mediated thrombocytopenia.
I'm very compassionate, by the way.
He is. He's the exception to the rule on every level.
Other questions?
I want to talk about the outcomes measure, right? So that's what I keep hearing. Sorry. I wanted to talk about the outcomes measure because that's what I keep hearing is just the lack of understanding of the disease burden, the patient-relevant outcomes, the mom who's the one that's on the front side of trying to understand how this disease is impacting, and I'd love to get your feedback from the pharma's side of it. How important was that outcomes measure data research that you did as part of your process?
I can speak from the consortium perspective. I think it's hugely important. Now that I am part of a biotech and part of a foundation and part of the outcome measure consortium, I can see it from all sides. It's the only thing the FDA cares about. And so you're doing this for drug approval, right? And so what does pharma want? They want to prove drugs, why it makes money. Okay, so let's work backwards. You need a drug approved to make money. You want to help patients too, but really money, no mission, right? So you need to make money. You need approvals. You approve based on efficacy. What is efficacy? You need to prove that something changed. If you don't understand the baseline of what you have, you don't know something's going to change. And so you need to then look at change, but not just from a parent's perspective, because I can give her, a piece of banana and it's a little bit too carby, and then I think that she's neurologically inappropriate, and that's probably placebo. So there's a lot of that. So then you need biomarkers that are like objective measures that can look at change with non-objective measures that feel like change. So you need outcome measures that feel good and biomarkers that look good. And both of those together is what drives up drug approvals. So it's hugely important. And I think that was one of the biggest drivers for pharmaceutical companies coming into our space. Every time we talk to them, and when we talk to the FDA about this consortium, they're blown away that such a rare disease has done all of this. And so it's much less work that the pharma has to do if we've done it for them. 100%.
And it's also about having that patient information as well and understanding what that looks like in the patient population, then looking about what that means to the caregiver as well. Our kids might all have a certain aspect, but are we interested in investing all our money in a drug going through to trial for that? Maybe not. So understanding what the parents' wants and needs are as well from the data that you've collected.
Yeah, you have to remember too that for a clinical trial, there has to be primary and secondary outcome measures, right? So if you pick the wrong primary outcome measure and you're a small pharmaceutical company that's doing this and you show negative results, it can be not only devastating to the clinical trial, but to the company itself. So finding really good outcome measures and really good biomarkers that they can hang their hat on and say, yes, our drug is making a difference and we have the data to show it. That's part of the battle, and that what she said earlier. I mean, it attracts even the smaller biotech companies and farm companies that want to come in and try their drug on a lot of different disorders, including Angelman syndrome, which we're starting to see too.
I just want to make a point that I spoke with Scott Gottlieb, the former FDA director, was at a value-based cancer conference. I'm from the cancer space, and he literally said, show me patient-reported outcomes data in your approvals, and that will help in expanded use. It's not necessarily creating that new molecule. That's all we all want. But expanding use and show me the data, and we will approve it. He said that Thursday of last week.
And don't think about it when you have promising drugs coming to trial too, because you're wanting to capture information over time. So if you don't have anything coming through the pipeline, it's never too early to start looking at a patient registry.
A question that I have, and I think the success stories we heard today are phenomenal, and I think that in part some of the success stories is because things aligned, and you've got some superstars that helped get you there. My question is, for an ultra-rare disorder, like the way that you went about de-risking to expedite the process, where can you find more information on that process to pick up on that? Because as an ultra-rare disorder with limited funding, time is the enemy. And for our kids, time is the enemy. And so I'd be interested in where we could find more information when we get to that point to try to shave off some time if we can. And I know it's not as easy as just saying that, but to find some more information on de-risking to be able to jump ahead in the process.
I don't know if that was a statement or a question, but I can comment on that certainly. I think I feel like FAST, and this is much of this is way before my time, paved a way for all of this. So it's kind of like somebody that invents something that takes a really long time, but now there's almost an algorithm. And at least from what I've seen and from what the FDA has shared with us is they feel like this is an algorithm that we should publish for other rare diseases. So we've spent and wasted a lot of time figuring out what works and what doesn't work. And now we've realized we see what works. And so I think that there's learning off of, someone mentioned this in the last talk, but learning off of other people's mistakes is huge. And learning off of other people's successes is bigger. And being able to partner with those other rare disease organizations to say, okay, we have like diseases. They're not the same, but they're similar. Let's have an outcome measure consortium because we all have nonverbal kids. We all have kids that have ataxia. We all have kids that have seizures. What are these like diseases we can put together and we can come up with assessments that are similar? That's more money and that's less time. So I think it's creating these networks of communities that are in these ultra-rare spaces especially, that are similar and can support each other. And I think that would save a tremendous amount of time. And you need one champion, not 4 champions. And this is another problem too, is the more people that want to control things, the harder it is to do anything. And that's one thing, is delegating people for jobs, because that's hard too.
And absolutely think global too, because us in the smaller countries, we want to be part of it as well. We don't need to do the science. There's a lot more capacity in the United States to do science than other countries, but we might have those skill sets in the smaller countries that you might need to bring to the table.
Hi, I'm Sue Hagan from the National Ataxia Foundation. Thank you for your talk. This was very helpful. We have no approved drugs for the ataxias, but there's a great deal of pharmaceutical interest just within the last 3 to 5 years. And so we're on the cusp of creating a pre-competitive pharmaceutical consortium. We've got a very strong clinical consortium. They work very well together, and they're collecting data, natural history data. And so I've reached out to all the pharmaceutical partners that have been contacting me and asking, what do they need from our community, from our scientists? They need patient-reported outcomes. They need MRI data. They need CSF. They need natural history. And so I'm trying to think of how I'm going to get all of our clinicians to start collecting all of this. And I had an aha moment in your presentation that maybe I just start with collecting one thing. Isn't that what you did with the biomarker collection? Or not?
Not exactly. I mean, we came up with priority lists, and then in natural history data or in the registry, it's asking questions. So there's retrospective data, and then there's prospective biomarker data. So for biomarker data, that takes a lot more time. Someone has to be willing to go under anesthesia for a TAP, in our case, maybe in your case, not anesthesia, but do an invasive procedure or a skin biopsy if it's a lysosomal storage disease. Like, those are some more invasive things. You could start with one thing, but if you have that patient at the bedside to get a biomarker sample, I would want to do as much as you can with that patient, and I wouldn't want to do one thing at a time, because I'd want to take advantage of that moment, because you don't have that opportunity too often. So that's where the natural history studies come in. Make a list of priorities in that study. What outcome assessments do you want? What testing do you want? What samples do you need? Create this biorepository where people can use that fluid, that tissue, or that 6-minute walk test, or the Actimyo, Actigraph, whatever it is that you're using, and everyone can then import that into a registry, whether that's a natural history study, truly, or a global registry in that it's more virtual. But in that situation, I might go bigger than smaller.
And if you were to look at perhaps, if you were going to perhaps look at building your own registry that was patient-entered, you would most definitely talk to the clinicians that are already collecting information and work out how you're going to connect that information. And then the beauty of having patient-entered is you can collect a lot of information in a short period of time. If you're looking at how many people can go into a clinic or how many people can even access a clinic, then you get to add to the data that they're collecting by doing something on your own sort of. But I would be talking with them about how this might look in terms of connecting the data so you're not having two separate silos of information.
We have some online questions too. So could you talk a little bit more about your mix of fundraising? It was, the question specifically is, what's the role of celebrity? How does storytelling and data collection interplay for you when you are strategizing your fundraising?
Want to take that?
Yeah, so celebrity is a big draw card to our annual event. I think, like Alison said, with Colin, the parents love to come and meet Colin. Colin loves to meet the kids. We have brought on, through one of our board members, is celebrity management, so he's been able to bring some different celebrities to the event as well, and it's helped us build the profile. The event in Chicago is, I think, one of the biggest events in Chicago held in the year, not just for a disease, one of the biggest events. We had about 1,100 people last year. People from all around the world. There's often a bunch of Australians that will come. They'll come from all parts of the globe. So that helps then spur global fundraising as well as in the US. So that's how we utilise the celebrities. And the second part of that question?
What are your, well, storytelling was a part of it. Is storytelling a part of why fundraising works for you? Are there other strategies for fundraising that you use besides celebrity and the big fundraisers?
I think that we do have some storytelling that I think is important and connecting. And I think for me, there's two ways you raise money. Right now, get people emotional, get them crying, get them excited, make it beautiful, give them a few drinks, and then do a paddle raise. So you raise a tremendous amount of money in an environment like that, and you invite guests that have means and are touched and involved in the lives of someone touched by that disorder. That's one way, and you raise a lot of money like that, and that's what New York City is really good at. Then, and now so is Chicago, but then there's also the storytelling, and it's the long, drawn-out fundraising. And so we have this thing called the CAN initiative, Cure Angelman Now. And what that is it's through Facebook. So Facebook has improved fundraising so tremendously, and it's improved just living with rare disease and creating communities internationally for rare disease when you don't live near these people, and creating friendships and decreasing depression probably because you're having these relationships that understand what you're going through. And through that, fundraising is really possible because your own networks, national and international, are on Facebook. And so we have this CAN initiative, which everyone creates their own little website, and you put it up on Facebook, and they can donate to that, which goes into FAST. And it's a story about your kid, maybe a video about your kid, and it's this testimony about why fundraising is important for your family. And then you put it up to your friends on Facebook, and they donate. And you can go and do a walk, or you could do a run, or a bike ride, or whatever it is, and everyone can donate to your CAN page. And then your own family, Quincy's Quest would be maybe my CAN page, and then I would have all of my friends donate to that. And then she raised $40,000 for FAST. And then you know what your family, what your network did, but it helps the bigger network. And it's the same mission and the same roadmap.
So that's CAN in the sense of able, not a can, right?
Yeah, Cure Angelman Now, CAN.
Okay.
Yeah, we call it the CAN page, yeah.
And they each organize their own separate fundraisers, or is this across the country you have a walk?
This is across the world.
Oh, across the world.
So this is just online, and people can just be like, or they can associate it with a real fundraiser, or they could just be like, it's Angelman Awareness Day, and I want all of my friends to donate $5 to my CAN page, and they put it up on Facebook, and at the end of the week, you have $2,000 that you would have never had if you didn't ask. So it's a really easy way to raise money.
Great.
And it's not GoFundMe, so they don't take money out. That's the nice thing about Facebook, is that they don't take a percentage away.
Great. Any more? Or I have another question online. Are there more in the audience? Oh, sorry. Excuse me. Oh, okay.
I just have a small question. It's probably going to sound kind of, I don't know, like I should have already gotten the point on this. But what do you guys mean when you guys are saying, like, you're champion of hope or whatever, or what you guys are looking for, like, your champion, or what is that supposed to mean?
So I think it means somebody who's the warrior mom or dad. It's that person who is going to champion the mission, the vision, that's going to take a lead in science, take a lead in networking and data, take a lead in fundraising. You need a person.
Okay. Yeah, it means that's what I thought that it meant, but I just wasn't positive. So like I guess that would be me in a sense. So I just wasn't sure if that's what you meant, like if it would be the person who's taking the action or the person that gave you the reason to take the action, I guess was kind of my confusion. Thank you.
From what you said this morning, I think you would totally be a champion in your condition.
Yeah.
2 more quick questions. When it comes to running a consortium, what does it look like to effectively manage that engagement?
Looks like bringing all the stakeholders to the table. Is that like?
When it comes to running a consortium, that's a hot mess.
Yeah.
That's what I'll say. Consortiums are difficult. They start off pre-competitive with all of the best intentions. Then someone gets into a clinical trial. One pharma company is no longer pre-competitive. Suddenly things become competitive. So it depends on the type of consortium. For scientists, you can speak to that. For the outcome measure and biomarker, it started off super supportive, and now it's because everyone's fighting for the first in human of whatever platform, it's becoming a little less so, and that's becoming difficult. So someone needs to be a manager of that for sure when money's involved. I think for science, it's
scientific consortia. It takes away all the competitiveness. So you're no longer competing against the next guy that's doing Angelman syndrome research for a particular funding, because you're all doing different things. You all have your own space, and you're all sharing data. So it's a perfect situation.
Interesting. Thank you. One last question, then we'll move on to the next session.
Hello. Is it on? Okay. Hi, I'm Tanisha Washington. I work for the Fibrous Dysplasia Foundation. I also teach at a university, and I'm a researcher. But outside of that, I really appreciate Global Genes, first of all, for the scholarships that they offer for people. I know we mentioned low income at one point, to be able to attend some of the events that you all have, and I also appreciate the conversation. One question and one thought that I do have, one of my main goals in rare disease space is to diversify, and I mean that as far as racial, ethnic, as well as income. And when it comes to that, I know you all mentioned that you actively do things for individuals that are low income. How does that community engagement look like for your organization?
I think you can speak to the global registry, because there's no, I think as long as you have internet, there is no limitation. And so for us, that is the same thing, I would say, from the foundational perspective. So everything is available online. If people can't, we have scholarships for people to come to the conferences. If they can't come still, because you can pay for someone to fly there and to their hotel and to be there, but who's going to pay to take care of their kid that has a significant disability that someone needs to be there with them? So sometimes it's just not possible, but everything is online live, so there's always access. And for us starting genetics, the main driver of that, besides having our finger on the pulse of timelines and safety and efficacy, is to ensure that no patient, regardless of who they are, what their last name is, where they're from, or what they look like, can get treatment. And so we want to be sure that no patient is left untreated. That's the most important thing to me. And so education and involvement and support through Facebook has been super easy for any ethnicity. I think the bigger issue with low socioeconomic and diversity is diagnosis, because it's really expensive to get diagnosed.
Right. And that was my next question.
That's a huge problem, because if you look at our population, the percentage, it's an equal opportunity destroyer, Angelman syndrome. So it affects randomly 1 in 15,000 individuals. It doesn't matter what color, sex, age, where you're from, what country, it doesn't matter. But we only have probably less than 5% of the Black population that's diagnosed with Angelman syndrome. And most of them most kids with Angelman that are in those populations are misdiagnosed with autism or cerebral palsy. And so it's a matter of, if we get an approved drug, getting it on the heel stick at birth. So having prenatal, it's now on prenatal screening, but then most people aren't going to take that next step to do an amnio because that's expensive. But if you can get it on the heel stick for, newborn screens, now
And
you're going to
they
be able to
have-
diagnose them much earlier,
Perfect.
and now everyone's going to have similar opportunities, and everyone is working toward that. But you can't get on a newborn screen until you have an approved drug.
Right.
And you can't get an approved drug until you know the population, and so it's this vicious cycle of how do you deal with that?
Yeah, no, thank you. That was my second question, because I was going to say most people can't afford to get a genetic test if they
Correct.
don't have the funds to do that. So thank you so much for that.
I think just really quickly from a data perspective as well, something I didn't mention, what we're trying to do is reach out to global patient organisations all around the world and looking at how they communicate it culturally sensitive to whichever country they're in. So making sure they don't always have to be on the internet, but they can connect through their patient organisations. I think that's an important message for everyone. Keep connecting with your global friends. Sorry to push that point again.
Thank you.